working behind the scenes to make this all happen. If you're new to the Digital Cafe, I'd like to just tell you maybe just who we are as a Soil Health Nexus. We're a group of university-led team dedicated to increasing the knowledge of soil health, science-based, research-based knowledge on soil health. And our team includes representatives from all the 12 North Central region states. So the format for today is our presenter, Dr. Anna Cates, is going to present, and then we're going to have Q&A. And if there's time, we'll go into breakout rooms, and you'll have the option to pick whether you want to go into researchers, educators, agency farmers, or students. So with that, I'd like to welcome Dr. Anna Cates, assistant professor from the Department of Soil, Water, and Climate at the University of Minnesota. And I'm going to stop sharing my screen and welcome, Anna. Good afternoon.
Thank you very much. Paul and I were just talking that it's so funny that he changed my title there from what I had originally reported that you might have seen on your webinar. And that tickled me because I was just about to explain that the talk title didn't actually fit what I was going to talk about very clearly, but Paul's adaptation kind of did. I'm essentially going to talk about soil health where I live in Minnesota, relevant to the upper Midwest, and some of my thoughts and efforts to improve our ability to quantify soil function. You can see a couple of my graduate students, Hava Blair and Bailey Tangen, listed there, as well as Jess Gutenecht and Marcel Lewandowski that I've worked with a lot on these ideas. Bailey and Hava are both going to be presenting at the Tri-Societies in November if you go to that event. I guess I'll also just say thanks to the Nexus for having me. I've really enjoyed being involved with the Nexus over the last few years. So if you aren't on our email list or haven't checked out the website, I think it's totally worth doing to get more resources on soil health from a pretty legitimate group. Okay, so here in Minnesota, how do we do soil health? I'd like to start with this definition from the NRCS. My title is the State Soil Health Specialist at the University of Minnesota Extension, and I'm with the Minnesota Office for Soil Health. So it keeps me honest to be sure I can say what I mean by that phrase, soil health. And I like this definition because the focus is really broad. It's not just about practices, but about the functions. And that makes it applicable both at a farm scale, where you might care about functions like yield and trafficability and nutrient supply to plants, and also to the landscape scale, where you might care about functions more like water storage across a whole watershed or nutrient retention and the filtration capacity of the soil. So then the question outstanding is really, how much is it possible to change these functions and the ecosystem that underlies the functions? I work in row crop agriculture, and at this point, that system has pretty enormous negative environmental effects, and our soil health systems are intended to reduce those by improving functions like water infiltration and retention, and by retaining nutrients and carbon. So the basic premise of my work and much of our work is that by changing agricultural management, we can change our soil properties and therefore functions or landscape level outcomes. But I think it's still kind of an open question of how far we can push changes and how big of an effect we can have on outcomes either on the farm or the landscape scale. So this figure kind of illustrates those connections up to the landscape scale. The thickness of the arrows in this figure illustrates how much researcher information we have on a given connection. So we have, including myself in this talk, focused a lot of energy on the relationships between land management and soil properties. And then there's also quite a bit of research I won't talk much about today between land management and edge of field metrics. And what's missing, and kind of a fascinating scientific question, is can we go directly from this soil property scale to the landscape scale outcomes? In this case, this is showing in-channel water, but you could probably look at other landscape scale outcomes also, maybe especially climate resilience. We kind of know in theory how this should work, right? Increasing infiltration to reduce runoff and flooding is a basic example, but there's not a lot of good empirical evidence. It's difficult to link those two scales. This figure is from a perspective piece that Marcel Lewandowski and I put together over the last couple of years. It's in a special issue of the Journal of Environmental Quality devoted to that connection between soil health and water quality. So I just want to acknowledge that my conversations with Marcel have really shaped how I think about this, and I encourage you to look at the issue for a lot of other good ideas around how do we connect soil to the outcomes that we care about most, in this case, in water quality. So for today, given that sort of broad need in the research, I'm going to essentially talk about two different sets of studies. One is some data for context. I'm going to share what I think of as kind of a classic soil health study we have with and without soil health, and we looked at changes in properties. So how do we do soil health in Minnesota, and how do those practices change soil health in Minnesota? And then this is going to lead me into some speculation. There are basically gaps and questions that this classic soil health approach leaves. And I'm going to present a couple of efforts to fill those gaps. They haven't been 100% successful. So I encourage you to pile on to adapt them. I'll try to leave time for questions or discussion at the end, and you can get in touch anytime over email. But I put these out there as potential ways to get more directly at the functions that farmers and policymakers are interested in. Okay, so these 5 principles are broadly expected to increase soil health or increase soil function in general. And I put them up there just to make sure we're all basically on the same page. We want to keep the soil covered, minimize disturbance, increase crop diversity, keep living roots in the ground, and integrate livestock. And those really have to do with protecting the soil structure, feeding the soil food web, and keeping nutrients in the ground. So when we think about applying those in the context of Minnesota, this map shows the upper Midwest by soil carbon content. So darker colors represent higher soil carbon content. And I just think it's so fun to look at this map. If I'm ever depressed, I can look at the map and think, at least I live in a really high organic matter area of the United States. And that's especially true here in the Des Moines lobe, our glacial soils that extend down to Iowa, and then in the Red River Valley here in the north. So there's a lot of row crops on these soils. And so I'm going to talk about cover crops here briefly, just to illustrate sort of the tensions with using soil health principles on this landscape in the upper Midwest specifically. So first we have this short growing season. In central Minnesota, in southwest Minnesota, we max out at about 140 frost-free days, whereas up in the central part of the state, we're at about 115. So this just means that the window is tight to grow cash crops, and the logistical piece tends to be kind of crammed into a short window. On top of that, these good soils mean that we have really high yield potential, as we do, across the Corn Belt. So we have farmers there in southern Minnesota who know that they could grow 250 bushel corn, and they're really loath to endanger that system in any way. Of course, there are people down here who are trying to go really high yield corn also, but they have a few more days to incorporate something like a cover crop compared to what we have here. And then certainly there are people up here who are also dealing with this short growing season, but they're no longer expecting to grow really high corn and soybean yields. They have more diverse systems, and they have less precipitation. So the yield potential is inherently lower. And so they tend to have a different ecological context and also different economic context. It's hard to find a place in southern Minnesota to sell wheat at a grain elevator. So it's difficult to incorporate a longer rotation and different crops compared to when you're up here where the whole system is kind of adapted to that. So we're working in Minnesota row crop systems. We have some kind of unique challenges, but we also have some really nice high organic matter soils. And I think it makes it a really interesting situation to evaluate this kind of classic soil health study, pairing conventional and soil health farms. So this is Hava Blair's PhD work. I think Hava is on today, so if I get any really hard questions, I'll probably pass them right over to her. But just to outline how this study is set up, it is all on-farm data collection. We worked with local partners to try to find farmers who were experts at soil health practices and made sure that the practices had been in place for at least 5 years. We also collected a lot of management data from those farms. I'll talk a little bit more about that later. And we're evaluating the surface soils over a couple of years of sampling. This is funded by a USDA CIG grant with these really fun collaborators to work with. So we picked 4 regions of Minnesota to focus on, and you can see that they're highlighted on the map and also on the soil texture triangle. The coarsest soils in Hava's study are sandy loams and loams that are located in central Minnesota. And then the finest textured soils are the clays and silty clays of the Red River Basin, the former Lake Agassiz lake bed. The southern Minnesota soils range in clay loams and silty clay loams generally. So because we know that not just region, but also more local topography makes a difference in terms of what your soil looks like and how much it can change, we also sampled at 2 hill slope positions. So you can see the experimental design here where for a given farm where we had soil health management, we sampled it at an upper hill slope position and at a lower hill slope position. Same thing at a conventional farm. There is a reference or undisturbed site that's part of this experiment that I'm not going to talk about any of the data from today because I want to focus on what the effective management is on changing soil health properties. And just to give you a little bit more information about what these different regions look like, I'm going to show you some pictures of the soil profiles for each part of the state. So in the Red River Valley, again, we have these silty clays, essentially high pH between 7.2 and 8. And in addition to corn and soybeans, we have wheat and oats and sugar beets in rotation. In the southeast part of the state, these are loams and silty clay loams. There's some canning vegetables and forages in these rotations. They're a little different, and the pH is a little under neutral, 5.6 to 6.7. In the central part of the state, I don't know if I said explicitly, the 2 profile pictures represented here are the upper and the lower hill slope. So this is where we see the most dramatic difference, where the lower hill slope really has this thicker, darker topsoil compared to the upper hill slope. And that is some of the most steep slope, as you'll see, will come up as we go through. And then in the southwest, again, the lower and upper hill slope positions there, and these are kind of in our middle texture range. And this is really the heart of corn soybean country with a little bit of alfalfa, or a little bit of oats rather, in that southwest area. So that's what the systems look like. These are the properties that we measured in terms of soil health. I think you'll see a lot of familiar ones if you read a lot of soil health literature. And I'm afraid I'm not going to go deeply into most of these indicators today. I'm going to focus mostly on these carbon pools and then the aggregate stability. So the carbon pools are microbial biomass carbon as measured by chloroform fumigation, potentially mineralizable carbon, which is a 24-hour incubation, permanganate oxidizable carbon, another brief essentially incubation, and then soil organic matter and total soil carbon as measured by combustion and loss on ignition. So those are some of the properties we're going to talk about. And the first analysis I'm going to show you is essentially what was the effect of treatment, hill slope, and region, as well as year of sampling on these soil health properties. So I'm going to start with these carbon pools. I'm not going to look at everything every time, but these are fairly representative of what we saw in terms of what we think of as labile carbon pools. So these are interaction plots showing the mean permanganate oxidizable carbon or soil organic matter with conventional and soil health on the x-axis, and then the colors here representing the different regions. And this was very surprising to me. We thought that soil health systems would increase carbon pools as they've been shown to generally across the US, but that's only happening in some regions for us. And the regions where it's not happening, we don't generally think of as very similar. One is the Southeast, the Southeast is where it is happening. And then in the Northwest, we just generally see a flat line between conventional and soil health, even though we're running at relatively very high organic matter. In these 2 central and southwest areas, we actually see a decline with soil health practices in 2 of the regions. The last piece of information that's on here is just that if the dots are filled in, that was a significant difference between conventional and soil health. And if it's open, then it's a non-significant difference. So POX, you can see, follows pretty similar patterns to soil organic matter, which is what other people have found with POX, that it's relatively aligned or correlated with soil organic matter as a whole. The last thing I'll just draw you attention to is that the relative difference between a conventional and a soil health system here is about 1% or a little bit less in terms of organic matter, or about 100 milligrams per kilogram POX. So try to pay attention to that, and we'll compare it to the hill slope difference in a couple slides. The other property I'm going to highlight here is the percent of soil and aggregates greater than 2 millimeters, so macro aggregates. And here we see a more consistent and positive response to our management treatment across regions of Minnesota. Generally, soil health systems increased macro aggregation. We generally think macro aggregates are a positive thing because they can hold water, and they allow for large pores between them to let water through. And they can also hold soil carbon and protect it from microbial degradation, kind of slowing the carbon cycle. When you look at our hill slope treatment factor, you see a more predictable story than with the soil health management. Essentially, with our two carbon properties, we see that the lower hill slope position on the left in these figures generally had higher values in those carbon pools than the upper. So that makes sense with how we think about how fine particles travel downhill with water or wind or gravity. And those fine particles tend to have more surfaces to hold organic matter. So this was much more along the lines of what we expected. I'll point out that we have much bigger differences on the hill slope gradient than we did on the soil health management gradient. So for soil health management, we saw a shift of just about 100 units of POXy, and here we see shifts of up to 400 in the case of the Southeast. Same thing with soil organic matter. Those some of the management shifts were more like this, just less than 0.5% of organic matter, and we see the potential for relatively larger differences due to the landscape positions. I'm just going to note, I see one question in the chat: how long did we apply the soil health management, which I think is relevant. These were all systems that had been in place for at least 5 years. They were supposed to be locally relevant. So we worked with a lot of conservation districts to pick out farmers that they knew had been doing this for a long time. And then you'll see how we incorporated that management data a little more in a minute. I also see the note about infiltration rates, and we will talk about that in just a minute. In terms of hill slope position effect on aggregates, this was much more neutral. We didn't really see much difference among the hill slope positions, except for in the Southeast. So in our next analysis, we wanted to get a little bit more specific about management. So we took 5 years of management data from these farms. We know their rotation, manure application dates and types. We know cover crop species. We know what equipment they use for tillage. And so Hava used that data to kind of interrogate whether some of the inconsistent effects of soil health were actually due to specifically one or another of the soil health principles being applied in different ways in different regions. So I'll show you what that actually looked like. So we built these models with continuous management variables based on the soil health principles. So one was the number of spring or fall seasons with living cover. So again, we had 5 years of management data, and this would essentially mean any time that you had something growing outside of the corn and soybean annual growing season. So a maximum of 10 over those 5 seasons. And it could have been a cover crop. It could have been a perennial like alfalfa in rotation, could have been a small grain that was already growing or what have you at that time of year. So that's getting at our principles of maximizing soil cover and living roots. The tillage one is a little bit, is pretty straightforward. We just added up the number of tillage passes, weighting the primary tillage passes more heavily than a secondary tillage pass. So for example, a disc ripper was weighted more heavily than a field cultivation pass. So this gets at whether they were minimizing soil disturbance. To get at diversity, we looked at the number of unique species. We just added these up, and we did look at some other ways of doing this, but we landed on just the number of total species and that biodiversity principle. And then we looked at the presence or absence of livestock in the system. We didn't have enough different livestock incorporation methods in order to make that a more broad continuous variable. So we just did this binary approach. So in these models, we also tried to account for our categorical region and landscape position variables with continuous variables. So we used the percent clay in the soil and growing degree days and mean annual precipitation to get at the regional differences, essentially. And we use this topographic position index to track that landscape position. And this spread out our regions as well, because where we had regions with more difference in topography, like the central part of the state, there's just a wider range of topographic position index. The topographic position index takes into account where a point is relative to all the points around it. And so in some place like the Red River Valley, where there's relatively little topography, you just get a smaller range in that variable. Okay, and then the last one was that we did continue to include year in the model when it was significant in categorical models. So you won't see it on every figure coming out. All right, so this is how I'm going to talk about this data. I'll try to orient you to the figures a little bit. This is just our original figure for context, because we're essentially looking at the same data in a different way. It's helpful, I think, to have them next to each other. Our response variable is still that percent of soil in macro aggregates. And we have our different parameters in the model over here on the y-axis. On top, we have the management ones, and below that, we have the essentially edaphic environmental properties. If a dot is colored, that means that it was a significant variable in the model. All of these numeric inputs were centered by subtracting their mean value and scaled by 2 standard deviations. So this allows us to compare directly the coefficients among the coefficients. And so just to walk you through the example here, for tillage, say, when all other variables are held at their mean, the estimate for tillage is -6. That means that when tillage changed by 2 standard deviations, the amount of macro aggregates in a sample went down by 6 percentage points. So when tillage went from low to high, aggregate stability went down by 6 percentage points. Another example here, when topography changed by 2 standard deviations, there was basically no change in the aggregate stability at all, which is the same as what we saw when we looked at the effect of hill slope position as a categorical variable on aggregate stability. On the contrary, something like mean annual precipitation or year had a big positive effect on the aggregates. So let's see, a couple of things to notice here. This mean annual precip one, that essentially divides southern and northern Minnesota. So if you look over here, you can see that our more northern and central sites have lower aggregate macro aggregate stability than our southern sites. And so these high precip sites are pulled out with our continuous model also. And then the last thing, I'm sure you're all noticing that year is the biggest effect on here. We had, 2 pretty different years between 2019 and 2020. This shows the average precipitation between 2019 and 2020. And that drier year had higher variable or higher numeric outcomes for a lot of our variables, as you will see. There's been a lot of work recently looking at how especially organic matter pools are changing by season, and our data really backs up that the conditions prior to sampling in a given season can really make a difference in what your outcome are. Okay, so now I'm going to look at these carbon pools. And so this gets a little bit more complicated. I'll remind you what these metrics are. Microbial biomass carbon as related to chloroform fumigation, potentially mineralizable carbon, that's a 24-hour incubation. This is POX, we've been looking at this. Total soil carbon is measured by combustion, and soil organic matter is measured by loss on ignition. Note that these 2 variables are log transformed, so you can't make the same sort of easy with your eyes assessment of how much they changed. Whereas with soil organic matter, you can see, oh, the maximum change based on one of these factors in the model was 1 or 2 percentage points. Okay. So what jumps out here? We see that high clay increases carbon and high topography decreases carbon. Oops, that's a similar story to what we saw in our categorical models, where lower areas and high clay soils have more organic matter and clay. Similarly, we have those higher values with high mean annual precipitation. That's showing higher values in southern Minnesota and higher values on 2020. I want to look more specifically at some of the management variables and see if we can tease out what's going on with why we see relationships with different management variables. So this variable n-species represents a number of species over time, and it's negatively related to a couple of our carbon pools. So why would we see a no or negative effect of diversity on some of these carbon pools? I thought this figure might help clear that up to some extent. Here we have as our x-axis the number of species over 5 years. This is just total planted species. And on the y-axis, we have our POX value. And what I think this shows is that we have some farmers, especially in our Northwest region, that were very ambitious with how many species they planted. But in Northwest Minnesota, they probably didn't get very good establishment or success with all of those species. And so we didn't see very significant effects on the soil. It looks like, if you squint, there might be a positive relationship here at the 0 to 5 level of number of species over 5 years, but it's not something we can take to the bank, if you will. And overall, I think what it says to me is that just having very high numbers of species by itself isn't going to probably boost your carbon. Okay, so what else is noticeable I want to pull out is the seasons of living cover. That's this top variable, and it has significant positive effects on 2 and sort of neutral and non-significantly positive effects on 1 other carbon pool. So similarly, this is a figure showing the relationship between POX and the proportion of shoulder seasons covered. I apologize, I don't think I explained what the size of dot meant in the last figure, and now I've switched it on you, but I'll explain it this time. So in this case, larger dots means more precipitation, and I did that because precipitation was another important factor in this model. So these larger dots here in the southeast represent the higher precip regions of Minnesota, and you can see most of our blue dots are pretty low in precipitation. In terms of the relationship between the amount of shoulder season covered and POX, we do see this slight positive relationship. The model was able to pick that out. I think this is like looking at those two together is interesting. We need not only that high diversity, but we also need to think about how much of the season is covered. And I think that the fact that we have this positive relationship with the shoulder season coverage really speaks to the importance of that living root and also just keeping the soil covered over time. So it might lead us to maybe emphasize having more of the ground covered for more of the year. And again, I know this is a little bit of an odd metric, the proportion of shoulder seasons covered. And by that, I mean fall or spring seasons outside of the annual season, annual crop growing season where there was something alive in the ground. Could have been a cover crop, could have been a perennial, or could have been small grain. Okay. The last thing I wanted to show you relative to interpreting these models is just acknowledging our kind of top predictors on all of them, which is similar to kind of what we looked at before, which is that clay here on our x-axis and POX have this really nice linear relationship. And then there's also a relationship between topography and POX, which is pretty strong. In this case, the dots are larger. When the dots are larger, that means that we're looking at upper hill slope positions, whereas these relatively lower ones are these smaller dots. And so like here in the red central area, you can see that the lower parts of the hill slope are higher in POX. So that leaves me and maybe some of you also in this somewhat of a predicament. What do I tell farmers about how their soil might change with management? And this is not an abstract question for me. I know this is literally a picture of me talking to farmers about soil 2 weeks ago. And in interpreting this data, I am a little bit at odds, right? It's my job to provide research-based information on soil health, and it's also my job to increase adoption of soil health. And in this case, the data don't show strong influence of the practices on soil health relative to the amount of difference that you had based on things that you can't change, your topography and your soil type. So I have this data set with relatively minimal soil changes, even in long-term on-farm samples. So you can't say, oh, they've only been in for 2 years, or oh, they're just on a research station. But what do I feel like I can say about what will make a difference? It still kind of looks like the soil health principles. Farmers across Minnesota do a lot of full width tillage. And so reminding farms that they can get similar yields and improve their soil aggregates with no-till or low tillage system is still a really important part of our extension mission. I also feel like this living cover piece, as I was saying, could be important. A number of species, just to a point. We have some more kind of nerdy soil work to assess why some of our labile carbon pools respond to the practices and some don't, and kind of to what extent diversity is helpful. This is just a list of what our kind of most sensitive indicators are, and I put those out next to ones recommended by the Soil Health Institute and by the meta-analysis from 2018 that looked at response of different indicators to cover crops. I feel like we're relatively in alignment with these. So sometimes I frame things for farmers in terms of nature versus nurture. Our data shows this really strong effect of nature, if you will, even when we really carefully paired up sites to try to account for all the edaphic effects. And so I'm, like I said, kind of in this pickle, and it's intensified by a study that Jess Gutnecht led over the last couple of years that I'm showing some data and some visuals from here. This was a really similar design in Minnesota. We had less careful pairing on soil type and landscape position and focused on just cover crops that were in an NRCS contract. And similarly, I'm not zooming in on this plot because all I really want you to notice is that for the most part, the lines overlap with 0, indicating that we didn't see big changes with cover crops in this study. But this word cloud is from a survey that we did of farmers in the study, and they overwhelmingly described soil improvements, most strikingly in physical properties. So there's something happening here. I'm not really completely satisfied with the word cloud or the anecdotal reports that soil is better, but I mean, it's hard to feel really excited about this really noisy data set that also is failing to measure something concrete. So what I'm really striving for now is I'm looking for a way to measure what farmers are experiencing and try to connect that to outcomes on the landscape scale. And this picture I find kind of inspiring in terms of thinking about what to measure and what's important. Looking at a flooded field like this kind of connects the field scale to the landscape scale. It's the situation that we most want to avoid at both scales. In the field, it destroys structure, it reduces yield, it's a complete and utter hassle. At the landscape scale, we're destroying infrastructure like culverts and roads, and we're delivering sediment and phosphorus to streams, and we see denitrification in a saturated system like this. So it's a goal for everyone to avoid this. And one way we frame that is in terms of resilience. I suspect I'm not the only person who's written a proposal in the last year that emphasized the resilience of a soil health system. And it's a difficult thing to assess resilience. My friend Greg Sanford in Wisconsin wrote this nice paper with a data set of a 30-year long-term cropping systems experiment where he showed that both diversity and perenniality could contribute to resilience. But for those of us who are too impatient to wait for 30 years worth of cropping systems data, we did something in Minnesota where we tried to stimulate those extreme conditions instead of wait for them. So we did this at some of the same sites that I presented in the first part of this. We just did it on a subset of sites because these rainfall simulators are kind of a pain in the butt to work with. So maybe some of you have worked with these before, but you can calibrate these sprinkler systems to deliver a specific amount of water to a plot. And down here at the end, there's an outlet where you can measure runoff. We're at 3 of the farm pairs from the first study, and we simulated a 50-year rainfall, which in this case was about 4 inches of rain over 2 hours. So this is a snapshot of an actual function, how much runoff, how much infiltration from these simulations. We also measured infiltration alongside doing it with the rainfall simulator using these Saturo dual head devices. So I'll present saturated conductivity or infiltration information, as well as the runoff and sediment loss from the rainfall simulation at these 3 farms. Okay, we found 3 sites that all responded really differently. So I'm going to walk you through them row by row. Each row of data here represents a different site, and each column represents a different measurement. So in the first row, we had a system with pretty low slope, and there was living cover in both systems, either oats or alfalfa in the conventional and the soil health system. You can see that when we look at the difference in infiltration rate, we found that soil health systems really did increase infiltration. But we didn't get any runoff or any sediment loss in either plot because we essentially had low slope and this living cover that slowed things down. So there, did soil health make a difference? Well, maybe, but it didn't kind of show the resilience to an extreme rain that you might want to see. And the next site, we had pretty moderate slope, 1 to 7%, and we had no living cover in either site. And in this case, we did see a slight boost in infiltration with the soil health. And we saw a fairly dramatic drop in runoff, basically with or without runoff when you have soil health or not, and therefore also a drop in sediment loss. On the last site, we had an area with a pretty significant slope, right around 10%, and again, no living cover. The soil health site still showed an uptick in infiltration, but runoff was pretty much the same or more in the soil health system. And the sediment loss was only a little bit less maybe in the soil health system. So here we can see that there's still this tension between how much the soil health management can change what's going to happen on the landscape based on the things you can't change, the topography and the soil type. So having living cover appears to be a pretty good idea, but also having a flat landscape is going to really help you prevent runoff. And you can't necessarily choose that in these systems. But this is kind of a cool way to actually show, like, you get this big rainstorm and what might happen. We'll have more data on this related to the nutrients lost in these that Jess Gutknecht and I are still working on. So back to our flooded field, the rainfall simulation suggested that soil health practices could somewhat improve infiltration, maybe not for this level of flood, but for some. And my next attempt to measure something really meaningful was to look at this anecdote of whether you can get into the field earlier with a soil health system. I hear this a lot from farmers. It seems like a form of farmer street cred to say, well, you know, it rained 4 inches and then the co-op could come out and spray my field, but they couldn't spray my neighbor's field. So that seems like something farmers love to talk about. And it is a really fascinating question. Again, getting to the heart of, like, what can we do about this flooded field? How much can we change our soil's response to this much water at a time? So in this case, I'm working with a master's student named Bailey Tangen, and we have 3 pairs of soil health and conventional farms. These ones, a couple of them have decades of reduced tillage in their systems on the fields we're measuring, and one is a little bit less, more like 5 to 7 years. And I'm going to test essentially a couple of hypotheses related to why you might be able to get into the field earlier. First, maybe your soil just dries out faster. Second, maybe you have more stable soil aggregates, and so you have a more resilient system because they're able to support wheel traffic even when wet. To figure that out, we did this kind of goofy sampling system, which has worked okay. I don't know if I would recommend it 10 of 10, but it's worked okay. Bailey's been out there about 24 hours before a rain event and about 24 hours after a rain event and a few days after a rain event to look at moisture and take a sample for soil aggregates. So again, these questions being, is it drier in the soil health system, and are the aggregates responding differently to rain Here's the little cartoon of an aggregate showing how this macro aggregate is held together with fungal hyphae and roots. Inside there, these micro aggregates, and it's the suspicion is that when you get a real soaking rain, that aggregate could dissolve and not be as strong anymore. So we'll see whether we're able to capture that. Okay, this is the moisture data. It's presented as volumetric water content. What percent of the total volume of soil is water at 10 centimeters and at 60 centimeters. So about 4 inches and 24 inches at our 3 different sites. You can see the soil health system is the blue. So we're not really seeing, these are 2021 data, a drought year. And we're not really seeing that there's much difference, if any, at the surface. If anything, the soil health system is a little ahead at one site. But then as you get deeper, there's definitely more moisture in that soil health system. In the 2021 drought year, that was probably a good thing for everybody, but there's no evidence that the systems are drying out more quickly when we're using soil health practices. So for our second question, is there more stability in these soil health systems? Here I'm showing again the 3 different sites and the pairs of farm, conventional, and soil health management systems. And I have 3 timings along this x-axis, but we're going to start with just that pre-rain timing. And the y-axis is the percent of the total soil mass in different aggregate classes. So aggregate stability was measured using a stack of sieves, and the soil was essentially partitioned into different size classes. The bar that's easiest to follow here, I think, is the dark red bar, which represents the largest aggregates, which are super sensitive to disturbance, and they're also important because they can protect organic matter inside and create possibility for infiltration around them. You can see that they're sensitive to management in this data set. In almost every case, the soil health management system has more large aggregates than the conventional system in every case. And that I did run a model on that this morning, and that was a statistically significant difference. Okay, so soil health increased large aggregates. Again, now what I really want to see in this is, can we actually assess how soaking rain might change how those aggregates respond? So this was after running a little infiltration test in the field at different sites, but this is kind of what I was expecting with the, sorry, there's some voice in there. This is what I was expecting with the conventional site, that the aggregates would dissolve and the whole system would kind of turn to jelly. Whereas over here, I would call these aggregates still intact, and there's still kind of air and flexibility in the system. However, we're not able to assess that with our aggregate stability method and our sampling timeline. They essentially, all the aggregates respond really similarly, and after a rainfall, aggregation doesn't change very much. So I don't think that this is going to be a great way to figure out what's going on after a rain and give farmers better understanding of why a given field might or might not better respond to rain. So just to answer my little hypotheses questions, no, the soil does not dry out faster. Yes, it is more stable, but it doesn't respond differently to rain. So I'm going to wrap up here pretty soon here. I feel like I don't have a perfect approach yet to capture the importance of this phenomena and how our soil responds to these phenomena, which in this climate change context is important to try to understand how we can respond to a big rain like this. My ideas are to make sure we're measuring things that farmers can see, like erosion and water infiltration. As an educator, it feels important to have things besides these cool organic matter pools that farmers can really latch on to more easily. And then I'm really interested also in continuing to understand the response to rain. If this wet aggregate stability measurement is maybe too coarse, maybe we look right at the surface of the soil where you get the most damage from a raindrop or that kind of thing. And then I also think that, things like the rainfall simulation and infiltration measurements need to be sort of zoomed out to landscape scale resilience. And I put the little smart aleck remark that this could perhaps be considered climate smart agriculture. I think that with the recent granting program from the USDA, we're really trying to figure out how to connect measurements we can do on the field scale to the landscape scale resilience. And so I'd love to have more conversation about that. And just to summarize what I've been showing with data, we do have some ability to alter our soil indicators and properties, but it really varies a lot with context, and the context seems to have the bigger effect. And then we just need to keep working on developing effective measurements to make the soil water connection relevant to both farmers and those policymakers. I will stop there. Thanks to all the fun people I get to work with in my lab, those rainfall simulation measurements and the sampling around rain events is pretty labor intensive. So I'm grateful to the fun people I get to work with.
So thank you very much, Anna. I know there's some questions in the chat that still need to be answered.
Yes. Yes.
And I'm not quite sure where to start.
Yes, Hava's been answering some of
Yeah.
them. I think I'm going to start kind of right under where Hava was chiming
Okay.
in. Jill Batchelor wants to know if there's any consideration of soil biota in metrics of soil health. Yes, there is. I guess I would say that I like those organic matter pools as metrics of soil health because they essentially affect all the biota because they talk about the food source for all the biota. I know that Christine Sprunger's lab and Michigan State is looking at nematodes as a way to assess the food web health more generally. And I think that that is really promising. But right now, for a lot of the things that we can measure, like microbial indicators like phospholipid fatty acids or PLFA, we don't know enough about what each of those groups do to be sure that it was an effective measurement. We can say there's more of them, but we don't know whether that'll change what happens. Okay. Does the dominance of intrinsic environmental conditions over soil health practices argue for better identification of a particular site's suitability for field crops and perhaps directing it towards different land uses? I love that idea. I think that it does essentially point towards the need to put these practices in place where they will be most effective. That's really a modeling question, Beth, that I haven't spent any time on, but I agree that it points in that direction. Okay, did I measure the effect that residue or cover crops had on aggregate stability? So that should have been inherent in both of the on-farm studies that we tested. I didn't go deeply into the management practices at our quote soil health farms, but they all had reduced tillage, which should have more residue, and many of them used cover crops as well. There's just this ongoing tension, right, between getting specifically at a metric like that or a specific practice and the whole system. And so both of these trials were looking more at the whole system of management rather than a specific practice. Someone asked if I think there would be more indicative results further south into Kansas, Oklahoma, and Nebraska. I think that when you have the soil health principles are intention, they're supposed to bring more photosynthesis into the system through more diversity and more living roots throughout the year. And I think when you have more carbon pumped into the system, as long as you're keeping that disturbance low, you should have more effects. It's a big issue in Minnesota that it's hard to get very much biomass from your cover crops. And so I do think that and some researchers have actually measured the relationship between biomass production and the amount of response you get in your soil, and it tends to be an important driver. So I think about that a lot in terms of when I think about my farmers and them implementing cover crops, did they implement them enough essentially? And how do I support them to implement them as effectively as possible? Have we tracked down any study that collected water content in continuously? Yes, I am doing that in one study, and I didn't mention how we did our water content in the field workability study I talked about, but that was just at the times that we visited the field. I do have some continuous sensors out there, and then there's a question of how deep you go into continuous soil monitoring. Aaron Day's lab, formerly at North Dakota, did some really cool work showing the temporal patterns of microbial communities in relation to temperature and moisture. So that's a really good reference on that topic, but it sure does get to be a lot of samples. What did I miss, Paul or Hava? Anyone want to point me up?
I think you've got them pretty well covered at this point. I guess what we kind of behind the scenes talked about, that we probably won't go into break rooms because the presentation has been so interesting, and there's been a lot of questions, and I guess I'd invite people to put questions in as they come up, as long as we have the expert mic'd up.
Yeah. I see that there's one in here about conducted interviews with participating
Yes.
farmers. And that is an ongoing thing. I didn't mention, so Hava put in the chat that we did in her study do interviews around management. And there's also some case studies with those farmers that kind of talk a little bit about more why they do what they do and a little bit more about economics. So there is a little more information on the non-biophysical side for that study. And then in the soil field workability study, we're trying to collect survey responses from a lot of Minnesota farmers in order to understand how important those extra days in the field might be, and also just see if we can get, a few dozen people to respond and tell us whether they actually do get in the field before their neighbors or not, or if that's just something we hear, like you hear the high number from the yield monitor.
I'd like to kind of weigh in on the trafficability thing that you talked about. And, that's very common in this area, in the farmers I work with that have multiple year, 2 decades of no-till, how they comment on the trafficability in the spring, and even at harvest time, how they're able to traffic. And it's something that I think is farmers see real. We're just not measuring it very well, I think. And that's more of a comment.
Yeah, and there's certainly, some big brother type approaches you could take with satellite data or what have you to see when different fields get planted. I would say with soil health, in the northern context, it's a little bit at odds with just the fact that soil with more residue is likely to warm up more slowly, and that might delay planting for the spring work period. But once again, people bring it up for harvest and for spraying also, not just for that spring period where you've got the competition with residue for soil warm up.
Claudia put a question in, did any of the attempted cover crops show an increase in biomass as compared to the others?
We didn't measure biomass of these specific cover crops. We weren't running any protocols to standardize management on the farmers' fields. And so the idea was really whether with these snapshot measurements, recognizing that they're limited by the effective season, was there any long-term effect of the years of management prior? So what could we say about the sort of long-term effects of these management systems, not just how effective cover crops were in a given season?
So are there any other questions? If not, we'll wait just a minute, and if individuals have questions to put them in, but I'd like to thank everyone for joining us today. It's really been a great presentation. I'm looking forward to reviewing some of those sections of the talk, because I find them very interesting. Join us next month, October 19th, where we're going to have Ateen Herrick from Dr. Jennifer Blesch's lab at the University of Michigan School for Environment and Sustainability. They're doing some really interesting work on cover crops in the Great Lakes. So I'd like to have everyone join us if you can next week. And if there are no other questions that have came in.
Paul,
yes.
Seth Await raised his hand.
Okay. Just unmute yourself and ask the question.
Yes, Anna, wanted to know if that infiltration rate data that you have, is that available for you to send to our office? I'm working with potential for reduction in flood downstream through FEMA and the Army Corps of Engineers.
Yeah, send me an email and we'll figure out what we can share. I'm sure it has to be anonymized and what have you, but Yeah, I'd love to see it applied. And you said you work on flood reduction, because I do think that that's an underexplored area of this, is trying to understand the economic impact for small communities.
Well, I will also, if everybody wants to, we actually have that on website. There has been a presentation that included the NRCS soil specialist, Army Corps of Engineers, Silver Jacket, and myself. I can put that link on.
Great.
Okay. There are a lot of comments of thank you coming into the chat of an excellent presentation, and I certainly concur. So I guess with that, if any other questions, just unmute yourself and ask them. We'll hang here for a minute. If not, thank you again, Anna. It's been great. We'll get this thing up on the website within the next couple of days. So if you want to re-listen to it, you're welcome to. So with that, we'll call it a day. And thanks for joining us again. We'll hope to see you next month.